enzyme. And we're recording it for posterity. Thank you, Aish. So it's really a chance to connect. It's quite informal. We try and have people talking about interesting projects that they're doing. And today we have two such people, Haneef, who's going to talk about protein immobilization, and Minette, who will be talking about using CRISPR in Cameroon for typhoid diagnostics. But it's really open. So we'll try and keep the talks reasonably short and have lots of time for discussion. And it's really just a chance to share projects that are ongoing and lessons learned from across a number of different institutions and different projects and goals. So at the end, we'll update a little bit on some future plans for improving things like the website and the forum to make it easier to find information and protocols and work together a bit more. But we'll leave that until the final stages of the call. So we won't do a full round of introductions because I think it will take up too much time. But while Haneef is starting to share his screen and prepare his presentation, if you want to introduce yourself in the chat, do feel free, just pop in your name, where you're from, why you're here, so that anyone who's new can get a sense of who's in the room. And then I'm sure more information will emerge during the Q&A. So I think, Haneef, you should be able to screen share. I believe Deborah is now our host, or Aish is the host, and can allow that to happen.
My screen is...
Yes, it's just loading. It's saying it started screen sharing, but we can't see it just yet.
No, I think.
Yes, there we go.
Yeah, thank you.
Brilliant.
First of all,
you're expecting 10 minutes, Haneef, or 15?
Hopefully, I do my best. And
okay,
10 minutes.
Right. So we'll try and give you a 10-minute warning. It's very difficult to alert speakers in Zoom because once you're screen sharing, it's really hard to get your attention. But we'll try and give a warning at 10 minutes just so that we keep time and have loads of time for discussion. That's the main thing. So over to you, Haneef, and thanks so much for joining us and offering to present. Thanks. Over to you.
Thank you very much, Jenny. Thank you for this opportunity. Reclaim from this opportunity. I'm going to present about what Jenny mentioned, that immobilization technology and its application in life sciences. So this is the brief introduction. Me currently I'm working as associate professor in the University of Turbat. That my research interest in enzyme production, enzyme characterization, kinetic study of enzymatic and microbial fuel cells, and immobilization. Skills is, as I mentioned, that enzyme is a purification protein, some sort of kinetic study of the enzymes, and I also involved the enzymatic maximum enzymatic activity by optimizing its chemical and physical parameter. I use immobilization technology, particularly encapsulation using biopolymer. So these are the basic introduction of me. I little bit share about this screen, some picture of my home country. I belong to Pakistan, and there's in Balochistan as well. There's the picture at the you can say left side is my University of Turbat. In center, there is the landscape of my homeland in Balochistan, and this is the coastal area. In last picture, in the left side, very down. This is me and my institution, my students. Basically, you can see the culture of our dresses of male and female as well. Let's just share with brief introduction. Now I start with immobilization. As I mentioned, that immobilization in confinement for localization, not only enzymes, as I mentioned in there, we can also localize the cell, microbial cells, and with the rotation of their biological activity. These are the basic methodology used for the immobilization. Binding to a support means use basically a different a binding procedure. You can use physical inception and you can use ionic interaction between the enzyme and your support. You also use cross-linking agent to covalently bond your enzyme to your support, and you can also use site specific, because covalent bonding is very good for the enzyme immobilization, but maybe you can lose the enzymatic activity due to conformational or structural change of the enzymes. and site-specific nowadays have been used by the different researcher to specifically bound these specific enzymes to a specific amino acid enzyme to a specific support. The cross-linking agent is very wonderful. Cross-linking method is very wonderful method in which nothing will do. You just cross-link one enzyme to another enzyme using cross-linking agent. You use a barrier to not disturb the enzyme structure. You can use a center barrier between the two enzymes. You also may not use for the enzyme. Encapsulation is very, you can say, wonderful method that never disturb the structure of the enzyme. You just return the enzyme into a polymeric structure, and you can use it from different biological activity for performance of biological activity. In this method, you can also use to encapsulate the cells to not only make a micro environment of a single cell of microbial, but you can also use multiple cell in a single bead and multiple bead. You can do microbial consortia. You can generate a good microbial consortia by using immobilization, this type of encapsulation immobilization technology. This is the some advantage I mentioned here is compared to free enzyme. It is easier to handle it enhance the stability of enzymes, the protein by protecting them by hydrolysis or heat or other organ solvent which is used in industrial process. A feces separation from the product. You know that enzyme is a very production enzyme is very costly. You have to use different substrate, different commercial substrate for the production that you can say cost a highly amount for that one. If you immobilize them in a support, make a reusable from different process. So you can cut off the cost, reduce the cost of production of that enzyme. Another very important that you just separate the enzyme, easily separate the enzyme your product. So downstreaming process, purification process is being reduced by using immobilization technology. This is the a control reaction. You can just not need any stopper of reaction. You just separate your enzyme from your substrate and stop the reaction. That's, as I mentioned, it is reusable, so maybe become more feasible for industrial application. This is my PhD research which I use for the in immobilizing technology. I use a very specific enzyme with sulfur pectinase. Pectinase is the enzyme which is used for the degradation of actin, which is part of the plant cell walls, biomass, and that's used for degradation. And its application is this enzyme application is in food industry, textile industry. Nowadays, it is being also used for the degradation of biomass-based material, just like lignocellulosic material from cell wall for the production of biofuel as well. So I immobilize the pectinase using three main support, that's calcium regenate, agar-agar, and polyphenol gel. I found that thermal stability of enzyme is being increased, reusability become very important, and I suggest in my research that it's very entrapment encapsulation, you can say, a method is very wonderful for the retention of catalytic activity of pectinase as well as to make it usable from different process. Now, currently, my interest of research is which is going on the synthesis of graphene-based enzymatic bioelectrode. You know that the energy source is going to be shifted into a different due to climate change or due to carbon emission. Researchers are trying to build alternative source of energy, which is climate lovely, climate resilient based method. So enzymatic bioelectrode is one basically one method of one research based method which is useful for generation of alternative source of energy. And this research, I basically trying to use a graphene. Graphene, you know that graphene is a material of 21st century that's highly electrochemical potential material. So if you use enzymatic graphene material, that maybe you're going to capable of increase the electricity production of these bioelectrode. So I just trying to both methods, that physical injection, encapsulation, and column bending. This is I just get opportunity. If I get opportunity, And I also get a work on site-specific immobilization method to do not disturb the enzyme activity. The bioenzymes, which are I'm suggesting glucose oxidases, glucose dehydrogenases, alcohol dehydrogenase, and catalysis. These are the one side of electrode, which is just utilize your a substrate like glucose and another material to flow the electron one sided. And by ketone enzyme like ligases, beryllium oxidizes, ascorbate oxidizes, and hydrogen 4 oxidizes are used for that one. That used for the convert of oxygen, which is formed by flow of electron into a water molecule. So the people are also searching that we use dehydrogenases, for example. That's also very important. Dehydrogenases. They need hydrogen molecule. Hydrogen basically is a hydrogen fuel as well. That's also people are considering in research. If you use immobilization technology, you can combine these enzymes in a single electrode and can be capable of performing the multiple type of reaction in a single moment. So that may be you can say very good for bioelectrode generation. Uh, Another I propose to proposing for my research, another research job, that's we can use these immobilization technology from a combined enzyme system for designing a combined enzyme system that composed of celluloses, glucosidases, xylinases, atactinases, and ligases for the degradation of a lignocellulosic material. That's mean a pre-treatment is pre-treatment treatment is currently a very costly method. You use a mechanical method. You use a high energetic energy method. Energy methods. That mean if you reduce these one into a biological method, that will be a goal. You can also use make an encapsulated microbial consortia from a mobilization technology, and you can also use these combined microbial system into a degradation of these lignocellulose material. So and plastic degradation is also I have proposed from my group that we can use a combined enzyme from the, using ligases, different peroxidases, hydrolases, and traces from analyze their effect on the plastic degradation because these enzymes is individually work and that one. If you try to set them into a single support and to work in a one substrate in different biochemical reaction, you esterases just remove the ester linkages between the plastics and maybe ligases remove the carbon-carbon bonding between the plastic. So these hydrolyzes maybe perform some sort of activated and same plastic. So they may be combinedly support one another from degradation of these activity. This is just a brief of my research and immobilization, which I'm talking about, which I am trying to utilize in different parameters. So thank you for listening me. So I think I'm in a time or earlier as well.
Great, thank you, Haneef.
Thank you.
So do people have any questions for Haneef on the radical graphene work there or anything around immobilization of enzymes? And I guess encapsulation of enzymes, which is not something that we've really discussed to date in the Reclone meetings. So yes, please go ahead with your questioning. If you'd like to ask a question, you can just unmute yourself and I'll keep an eye because there's not that many of us. So
hi, it's Keith from Manila. Could you describe a little bit about the plastic digestion, slide 9? I mean, specifically, I suppose, what state of readiness it is. I don't know about any of these enzymes at all. So is this research level in production? You just expand on that, please. Thank you.
This is basically people. I just give one example of lacase. Lacase is currently discovered from is a phytase enzyme, polyethylene tetraethyl degradation. Lacase is esterases. These are different enzymes which is on working on these plastic degradation process. That's not mean that not that's depend on the type of composition of plastic, what type of that. Which currently they are not quite applicable in the large mass level. They are just initially going to be kept doing that. The fungal is one story. They use microbial degradation of plastic is one story, but microbial use the same sort of enzymes which is degradation of the plastic. So this initial, and I just propose the combination of these combo of these enzymes maybe get better effect on the plastic degradation. So I think I maybe I'm answered your question.
Yes, thank you.
You're welcome.
Has anybody else used some of these techniques in their own work? I mean, we've not done any encapsulation, but I think I'm really interested in particularly some of the hydrogel-based encapsulations, because they're quite attractive in terms of requiring quite low cost and sustainable resources that could, really be quite helpful in a scaled up context. We're always trying to think about circular material flows in some of our protocols, and those kind of materials have a really good opportunities there, I think.
That's very interesting. When I start working of encapsulation of enzyme using calcium alginate leads, so we use a sodium alginate is the form of solution. I just mix up the enzymes with this sodium alginate solution, and use a needle, just a syringe needle we use from medical purposes. So I fill up this syringe needle with enzymes and my alginate solution and drop into a chloride solution. So when I drop one single drop into a calcium chloride solution, what happened? The sodium is 1 plus charge and calcium is a plus 2 charge. So more affinity with alginate is compared to sodium. So when they touch the calcium in a solution, the alginate replace this sodium with the calcium. They form a calcium alginate, and calcium alginate in the always form in the solid form. They come into a very fantastic beads like it. When I check to how the substrate going to be reached to inside of these encapsulated enzymes. So there is a very important theory about the diffusion. When we separate, we use a barrier the polymer of alginate, and the barrier have different foresize. They are different. If the form of solution, substrates try to flow from outside solution inside the solution due to the diffusion. When they come to diffusion into inside, the enzymatic reaction going to occur, and the substrate, whatever the substrate is going to form a product. Product is very less in the outer environment into a solution. So product will starting to flow outside in diffusion form. Whenever you reach equilibrium condition, whenever that may be not possible, because when the substrates are gradually is degradation. So at the moment maybe equilibrium going to be reached, now the activity going to be stopped at that moment. But one important factor is you can use a single reaction. You can of a single process you can perform multiple reaction with the combination of first two second reaction. For example, if I use one microbial just cyanobacteria to produce a formic acid from carbon dioxide. So formic acid is basically substrate from yeast. If you use a yeast in single strategy, they will formic acid will be converted into another product. That's for example 3-hydroxide for 3HP. So 3HP is very good components from using biodegradable plastic formation and polymer science. So you can do that combination of different. You not need to purify one product from the reaction of another product. You can just use it in single reaction mechanism, in single reaction mixture for multiple reaction which you are interested to do that.
Great, thank you very much. Oh, so we've got a question from Scott, who's been looking at a paper on Twitter. Always a good start to a conversation. What's your question, Scott?
So yeah, excuse me. I was just having a look at that paper, and I was thinking, you know, when you're immobilizing things, you're impacting its catalytic activity, and I guess if you can produce enough of it, you can just keep refeeding that. But I'm wondering if there's some ways to make the activity of the enzymes stable for longer while they're in the immobilization context.
Yes, as I mentioned that two things. If immobilization always, if you're doing in a polymeric reaction, encapsulated beads. So one of the major inhibition of enzymatic activity is substrate product inhibition. For example, if I use a amylase for degradation of starch and produce glucose itself is a control-based inhibition of amylases if the concentration are going to be rise in a solution form. But if you use the encapsulated beads to restrict the enzyme into a microenvironment, then when the glucose produced, glucose start to flow out of the microenvironment due to diffusion of concentration, because hot environment starch concentration is higher, so glucose try to flow outside, and this is also maybe increase the longer base stability of enzymes. If you about the other ionic strength of the solution, maybe you can it's stable because there is restriction, a barrier of polymer. Polymer maybe restrict the thermal effect on enzyme. Maybe enzyme going to be a higher thermal stable compared to soluble form, as well as there may be operational stability of enzyme going to be increased because that resist the thermal as well as the ionic concentration of the solution which you are using in it, and this product-based inhibition is also going to be reduced. I think I answered you correctly.
Yeah, thank you.
Welcome, Scott. Welcome.
Great, thank you, Haneef. So we're going to move on to hear Minette's talk now, but if you have any questions for Haneef, you can feel free to pop them in the chat. And if you wouldn't mind stopping screen sharing, we will move to Minette, who can take over the screen from you.
Okay. Hello, Jenny. Hi, everyone. I'll share screen in a minute. Okay.
Great, thank you very much.
All right.
Lovely. All right, thanks, Minette. Over to you.
Okay. So I will be presenting on the local production and validation of a CRISPR-based typhoid diagnostic through a novel academic community partnership. What is typhoid fever? It's a bacterial infection caused by Salmonella typhi and to a lesser extent, Cerovascular typhi A, B, and C. Typhoid is potentially fatal, multisystemic disease, and without prompt treatment, it can cause serious complications. Its transmission is mainly through the fecal-oral route via contaminated food and water, and it is diagnosed mostly using serological tests or culture of bone marrow, blood, and stool. It's treated using antibiotics, but its clinical management is currently being challenged by limitations with its diagnosis and antibiotic resistance. I would like you to meet Rosa, sorry. Rosa is a food vendor who lives in a rural area in Cameroon with a history of typhoid fever. She visits the hospital presenting clinical symptoms, signs and symptoms of typhoid fever again. So these symptoms are non-specific. These symptoms are non-specific and requires a diagnostic diagnosis using a diagnostic method for confirmation. In Cameroon, mostly serological methods are employed. The cultures are reserved for standards. In which case, if the doctor requests a serological method and what is used in Cameroon is without, it's prone to false positives, and it's prone to false positives it's prone to false positives in patients with history of typhoid fever infection. So in which case, if Rosa is diagnosed positive, it could be a false positive or resistance, and she requires a confirmatory test, a confirmatory culture test to determine if it's actually typhoid fever or a case of antibiotic resistance. But most rural areas lack the facilities to perform culture. And if she has to travel to get to a town, perhaps to get the test done, it's costly and time consuming because it takes about 4 to 7 days to get the results. So in which case, so in most cases, Rosa will probably prescribe an antibiotic and be sent back into the community. And this is a risk to herself and to the people around her. Thus, typhoid fever faces the challenge. Typhoid fever remains a public health challenge due to limitations in its diagnostic tests, lack of minimum laboratory requirement for culture tests, and emergence of resistance. The global burden of typhoid fever, there are about 11.9 to 26.7 million cases worldwide, 80% of which occur in low and middle income countries. An estimated over 12,000 to 200,000 deaths occur each year too. 45,000 cases and 580 deaths were recorded in Cameroon in 2016. 70% of these were children aged 1 to 15 years old. Typhoid fever. A main problem with handling typhoid fever is antibiotic resistance. Early on from 1948 to 1970, antibiotics chloramphenicol, ampicillin, and cotrimazole were introduced as effective treatment for the disease. But by 1980, multi-drug resistance emerged. Fluoroquinolones, multi-drug resistance emerged, which is resistance to the first-line antimicrobials, chloramphenicol, ampicillin, and cotrimazole. Fluoroquinolones and cephalosporins were then introduced, but currently we are dealing with extensively drug-resistant bacteria. This bacteria is resistant to all the antibiotics recommended for treatment except macrolides and carbapenems. So CDC reports an ongoing outbreak of extensively drug-resistant typhoid fever, which has taken 5,000 people in Pakistan and 11 children in the United States who traveled to or from Pakistan. So it's actually very big issue. There is therefore need for rapid and accurate diagnosis of typhoid fever, generation of data on the prevalence of typhoid fever and antimicrobial resistance, especially in Cameroon where this data is lacking. The general objective of this study is to develop and validate a proof of principle, low-cost DNA-based diagnostic tool for typhoid fever using the CRISPR technology. This tool will be capable of distinguishing Salmonella typhi from paratyphi infections and detecting antimicrobial resistant genes in the bacteria. The specific objective of this study is to determine the prevalence of typhoid fever in selected health facilities in the Bako division of Cameroon Southwest region, and compare sensitivity, specificity, positive predictive value, negative predictive value of without and Cas12-based diagnostic relative to the PCR. Then determine and also to determine the prevalence of antibiotic resistant genes in the selected health facilities. So all ethical requirements taken into consideration, the study population includes individuals reporting to target hospitals demonstrating the clinical features of enteric fever as confirmed by a medical doctor. The sample size is 410 participants. It was calculated based on the previous study in the Northwest region of Cameroon. The sample is whole blood, 3 to 5 ml for comparative analysis and resistance typing. For inclusion criteria, the patients demonstrating the clinical features of enteric fever or typhoid fever will be enrolled, and they must have a signed informed consent form. And patients without the signed informed or without informed consent can't be included in the study. So this study will merge CRISPR with an isothermal amplification to improve the sensitivity of the study. And as mentioned above, we proceed to comparative analysis and anti-resistance and typhoid resistance typing in the patient. So I'd like to show a simulation of the diagnostic process. So isothermal amplification of the target region will improve sensitivity of the assays I mentioned earlier. And Cas12 itself is an enzyme guided by RNA to recognize specific DNA sequences. Upon recognition of this sequence, the target is cleaved with collateral cleavage of a quench oligonucleotide, resulting in a fluorescent signal. And if you're wondering why CRISPR, well, this has been done before. The Shalo platform was quite successful. The Shalo platform has been successful in detecting the Zika virus and various pathogenic bacteria and single nucleotide polymorphisms, and is also based on the CRISPR module combined with an isothermal amplification method. A typical single plex reaction for the Shalo platform can cost less than $1. So it's appropriate, at least appropriate cost according to costs in the target area. DetectR, another platform too, has proven successful and has been used to detect the human papilloma virus. Cas12 too can use multiple guide RNAs for creating simultaneous double-stranded breaks at different target loci in the genome. This allows us to design multiplex assays in order to detect more than one specific DNA sequence. So for this study, strains and gene target. For antimicrobial resistant genes, we focused on fluoroquinolones and cephalosporins because those genes resistant to fluoroquinolones and cephalosporins because those are the major antibiotics currently used in Cameroon. The progress of this work, well, we've successfully completed sample collection and we've successfully completed sample collection alongside the serological testing, from which we're able to get a prevalence based on the without, which is clicked with several limitations, but the prevalence of over 60% was obtained in the FACO region. Currently, we are working on DNA extraction and detection of the bacteria by PCR. Also, expression and purification of the enzyme required for the Cas12 diagnostic has had some measure of success, but some optimization is currently going on. The expected benefits of this study. So awareness. So whilst collecting samples, there was the requirement of letting the participants understand what typhoid fever is, how it's transmitted, and debunking several myths that are common here about typhoid fever. So the some awareness was created about typhoid fever and antimicrobial resistance. The study will also provide data on the prevalence of typhoid fever and antimicrobial resistant genes in the selected hospitals. This data is quite important for future health decisions. The novel diagnostic tool. The novel diagnostic has a potential to be specific and sensitive, can improve clinical management of the disease and reduce the greater than 500 deaths that occurred occur yearly. It also is a form of local innovation because successfully introducing this technique in Cameroon opens up potential for further projects or expansion to other diseases that are prominent here in Cameroon. Thank you.
Can I
Great,
ask
thank
a
you.
question?
Go ahead, Guy, yeah.
Or what? Okay. Do you have any idea how many of the sort of antimicrobial resistance are actually SNPs or a whole gene, or do you have any feeling for that?
Yeah, with fluoroquinolone resistance, there's it's mainly conferred by a SNP, in the gene. But for cephalosporins, it's mostly the presence of plasmid that confers the resistance.
And then about the whole CRISPR thing, so from what I understand, also the gRNAs are quite expensive. And also all this CRISPR stuff has a lot of intellectual property around it. Do you have sort of any thoughts or what? How do you get the gRNAs or plan to and things like that?
Well, for reducing costs, we actually focus on producing at least one of the enzymes in the lab here.
The gRNAs are coming from Cambridge right now, is the short answer.
Okay.
But I mean, Minette's kind of
on dry ice? Or?
Okay. We actually had to ship them. I've forgotten how we shipped them because I didn't package them. But
Probably
we don't.
Don't.
Yeah, I don't think we sent dry ice.
Okay.
So there must have been it must have been a -20 shipment, I think. But Smitta isn't on the call today. She was the one who packaged it. We very, very rarely send dry ice to Cameroon because it's a right pain. So I think there was some formulation whereby we could ship it not on dry ice. But Minette has produced, I mean, Minette left out, I think, a bit of a presentation here because she has produced Cas12 already in Cameroon. And in fact, all of the enzymes necessary to do RPA, but we're moving away from RPA for other reasons. I think we had a conversation about RPA at a previous meeting. So yeah, the gRNA is a challenge. But Minette, maybe you could say a little bit about the need for sensitivity in terms of typhoid testing, or the high sensitivity requirements, I guess.
Yes, the thing is, sensitivity is quite important for the diagnostic because the bacteria is present in low concentrations in blood, meaning with blood culture, you can easily miss out the bacteria. And with stool culture, it's easily lost because it's not competitive enough amongst the other bacteria that are common in the gut. So sensitivity is quite a very high requirement for diagnosis and necessary. CRISPR, of course, offers that, especially since it's coupled with isothermal amplification and can reduce detection levels to attomolar quantities.
Can you talk anything about the expression work that you did to make the Cas? It sounds from Jenny like you've done it. Can you tell us anything about the plasmids or any problems or challenges? What worked well, yields, anything about that?
With Cas12, well, yes, there are a lot of challenges that we have, especially structural challenges. So I did express the enzyme and purified, and yes, the challenge came with testing with the quench fluorophores, and also at the level of purification, because also at the level of purification, trying to acclimatize the protocol to our environment here, and also get them working concentrations of the protein.
Was it a one-step column or two-step or three-step? How many cycles of purification did you have to go through to get the Cas?
So we compared, what we did was we are comparing histac purification to ammonium sulfate, and with histac it's just one cycle. It's just one cycle, and we don't currently have the results on which purification method is best yet, but we are hoping on histac purification, and also hoping that one cycle is enough.
Sure. Yeah.
It looked pretty pure on the gel, though.
As a
Would you agree,
general
Minette?
rule, sorry.
Sorry, carry on, Keith.
As a general rule, I noticed Fernand's on the call, and I think you do just one his tag step, but then there are other papers that define a his tag, and then I think an ion exchange, and then gel filtration. And for all the proteins that people have made, who have much more experience than we do, I'm just wondering if there is a collective wisdom as to whether or not polishing with the second two columns is in general needed, or is that such a broad statement that it depends on each individual case?
It's a really good question, Keith. Yeah, we haven't done any ion exchange or polishing steps, but we're about to have some reagents shipped to Cameroon to do just that. That's more for an RT lamp project, but we could, once the reagents are in place, we could equally try doing some Cas12. I mean, I think we've looked into it. You can get reasonably inexpensive ion exchange, but we don't have an actor. I know in Fernand, you've got HPLC and sort of equipment in Chile, and we don't in Cameroon. So we've been looking for stuff that we can do with just peristaltic pump or syringe driven. And there are some small ion exchange columns, so it's definitely an step. One of the things we realized with RPA is that probably stuff needs a bit of polishing, because that was challenging. The Cas12, we're not sure yet. It's been working in Cambridge with just a one his column purification without polishing. It's been working perfectly fine. It's unfortunate Smita isn't on the call, because she could probably show you some graphs, but yeah, so as Minette said, the challenge we've had in validating it in Cameroon has not been related to the Cas12, it's been related to a bunch of other stuff going wrong. So there's another parcel that's leaving this week, which should contain the things that we need to really nail it down. But yeah, Fernand, are you adding polishing steps on your enzyme prep in Chile?
Hi, Jenny. Not the ACTA, as far as I know, yet. Sometimes we do heparin columns for DNA binding proteins, but that's it.
Right.
And you're using this Amicon to sort of concentrate and clean out other stuff.
Yes, but not like ion exchange or any other step.
I suppose I'm just interested to know whether we have contaminating nucleases, which I suppose is my paranoia, and wonder whether that's the reason why you polish, but apparently not, from what people are saying. It's functionally good enough with one, maybe two columns, I think is the general lesson I'm hearing. Is that right?
Yeah, well, it seems to, we've got it working, but I think the nuclease contamination is an issue. So we're trying to test everything for nuclease contamination to see what is there. The one thing we have found is not successful. We've not managed to successfully get it to work without, we're going to have to add some polishing steps, is Clenal. For a sort of cheap Gibson, you can just use one enzyme, which is the Clenal fragment, and that doesn't work because it needs to be a super clean mixture. Basically, if there's any DNTPs or kind of other anything in the buffer that's carried over from the purification process, it interferes because you need to be completely DNATP free to get the Clenal exonuclease function to work. And then the exonuclease releases DNTPs, which turns it back into a polymerase effectively. so that seems to be very, very sensitive to really, really pure prep. And we've expressed it fine, but we can't get it to function. And we think that's the reason why. But other ones we haven't had any issues. Has anyone else found one that they need a polishing step for? I think the default is everyone tries it with a polishing step first if they're professional protein people. So sometimes it's difficult to tell.
But for the typhoid, it sounds like you're moving to LAMP now.
Yeah, okay. It's just so difficult to optimize RPA. There are too many parameters. So yeah, Smith is well. So Minette's focused on kind of at the moment getting the Cas12 working with. We have a commercial RPA kit, which we're just sort of focusing in on one thing at a time at the moment. I mean, Minette, if you want to sort of outline the priorities for the work for the next couple of months. I'm hijacking your Q&A session here, so you should take over.
So for the next couple of months, we are prioritizing, first of all, production of Cas12 and testing it against Twist TX RPA to ensure that it's functional. Twist RPA amplified fragments to ensure that it's functional. And also, next we'll prioritize expressing BST for LAMP. So
I can share a few interesting papers about doing like SNP stuff, new interesting ways of doing SNP stuff with LAMP, because I think there are better ways than the Cas12. So.
Yes, I guess that would be great.
Great, thanks. All right, Robson's saying that they're also producing Cas12 and Cas9 using iMac with I guess no polishing step, sounds like. So good to know that. I think a lot of people in the room have an interest in protein purification. So if anybody's got any kind of good tips that they've used, I see China's referencing in the chat syringe pumps. Yeah, I mean you can certainly get syringe pump driven columns. And the ion exchange, I mean, as I say, the media for that is reusable and is not too expensive. I think that the more problematic one for sort of supply chains and not being able to reuse it is size exclusion chromatography, because typically speaking, you kind of do want the commercial columns. They're quite difficult to make yourself, whereas the ion exchange, you can kind of get away with it if you try. And there's some interesting papers around kind of producing monolithic. So some monoliths of different materials. Of course, there's hydrogels as well to kind of link the two talks together. There's a lot of use of hydrogels for different ion exchange kind of functions. I was recently looking up purification of lysozyme, and there's some quite nice papers using like really easy hydrogels for that. But I think a lysozyme is super easy to purify by ion exchange because it's got an isoelectric point of about 9.5, which is far away from almost anything else in egg whites. So you can get some really clear separation. Cool. Right, guys, got to go before there's a lockdown here in Paris. So good luck with good luck with that. We're 5 minutes away. Is there anything anyone else would like to share in terms of work that they're doing that's related to the work we've heard from Minette or Haneef? Sounds like not. So yeah, I can because we're short on time, what I propose I send the I send my recent digging around on ion exchange resins to the forum, so that you have the reference links and we can continue there. Yeah, and definitely, Robson, if you have some information about RNA guide generation, the forum would definitely be a good place to discuss that. If people have got good ways that they've done that, and it's been easier than going through some of the commercial providers, then for sure that would be a really interesting topic. So in the last couple of minutes, just to say, please take some of these discussions to the forum, and also that we're working to develop a development plan for the forum to make it easier to navigate and better for people. So that will be in conjunction, hopefully, with Pavilion, who are a really nice non-profit developer collective who specifically work on designing. They basically work with Discourse, which is the forum software that we use, and they're big open source developers in the Discourse community, and their kind of specialism is really community building, online community building websites. And so we're excited that they will be able to help us out. So if you're interested in making the forum a really useful resource, then definitely mention in the chat or reach out to us, because we'll start a little working group to get that moving forward. On which note, in the last few moments, I'd like to introduce Deborah, who those of you who've been on the last few calls will know Kayla, who was a coordinator at the University of Cambridge who is spending some of her time helping this community. And Kayla has moved on to a new role, which we're all very excited about. She got offered an excellent opportunity with Cambridge Enterprise. So she's actually just moved a few buildings down the road, if we were even in our buildings right now. So obviously most of us are working from home. But yeah, so Deborah has stepped into the breach, and you'll be hearing from her. I don't know, Deborah, if you want to say a few words of hello.
Hi everyone, good evening, and I really want to apologize for all the many emails I sent out today, and I think Jenny said it's a mix of handing over and time change. So hopefully going forward, things you get more direct emails and less drama. Thank you so much.
Thanks, Deborah. Yes, today was the sort of the culmination of those two things. We will be on a solid track by next week. And also Aisha's on holiday in Alaska, who normally organises the meetings as well. So everything came at once. But we're delighted so many of you are able to join. And yeah, you'll be hearing from Deborah in the near future, who also has some great contacts from her own work, and maybe she can talk a little bit about her background when she introduces herself on the forum as well. We've got 1 minute to go, so I will suggest that we wrap up with just a thank you to everybody, and especially to our speakers, Haneef and Minette. We will be back again in 2 weeks' time. If you would like to present at one of these meetings, then do get in touch. I believe, although I haven't been CC'd into the emails, so forgive me if this is presumptuous, but I think, Emmanuel, you're down for presenting in 2 weeks' time, is that right?
Indeed, I think that should be the plan.
Excellent. I thought it was, but then I doubted myself for a moment. So yeah, so would you like to give a kind of very brief, with 1 minute to go, just what are you going to be telling us about in 2 weeks' time to pique people's interest?
So I'm going to give a very brief outline of our testing in several African countries of a LAMP kit for COVID-19, where we have done field work in Cameroon, Nigeria, Kenya, and Ethiopia, testing side by side this LAMP kit together with the traditional RT-qPCR to see if it was a more reasonable solution for testing for this virus in these countries. So I will tell you a little bit how the idea came about, as a preview of the results, which have been very positive, and I hope it will be of interest.
That sounds great. Well, I think it will be a lot interest to a lot of people in the room. I mean, particularly to call out, I mean, Minette and the rest of the team at Moboa Lab are working with a number of Cameroonian institutions on an RT-LAMP project right now. Brooke, the Ethiopian Biotechnology Institute, is also working on an RT-LAMP. So I think they're working more on the local production side, but it would really nicely tie it together to show that it works with the kits and then, you know, branch to local production. And Emmanuel and I are both on the local production and diagnostics working group, which is a sort of WHO, UN agencies kind of thing. we sort of spun off. But so we both, Emmanuel will not be presenting on manufacturing or local production, but he has an interest in that space, and the results will be very relevant. We've gone actually 1 minute over. So thank you so much. We will save the chat, and we have the session recorded, so we'll post the recording. And we'll make sure that is available on the forum. And please take these amazing discussions and tips and paper sharing over to forum.reclone.org. And we look forward to seeing you in 2 weeks' time. Bye, everybody. Thanks so much. Speak to you soon. So Deborah, I think you and I should both get an email about the recording, because it should go to the cloud.
Okay, and maybe I can stop.
Yeah, I think you can stop recording. Oh no, actually no, because we're in iShiz, so it won't go to our cloud, it will go to her cloud.
Yeah, okay. Yeah, of course. Let's stop it.